Hello, everyone, and welcome to Chapter 10. This will be part one of two. I really had strongly thought about doing this in three parts, but just time and things like that, the semester, just some things that led to me not really being able to do that. But that was kind of a hope that I originally had because the last chapter is kind of an easier chapter. So, what we're going to do is I want to remind you that opportunities are usually disguised as hard work, so most people don't recognize them. So we're going to get started with chapter 10. These are the things we need to know for this chapter, of course, that are required to be talked about. These are the things that will be on the test for sure. This kind of acts as a good checklist for your study. Can you do this? Can you do this verbally? Can you go out there and actually compare some of this stuff here? Now, we're going to begin just a little bit about muscle tissue and just remind yourselves that there are three types of muscle: skeletal muscle, cardiac muscle, and smooth muscle. And these are a little bit, they have differences histologically that we talked about in the histology section back at the beginning of the semester. And so when we talked about that, we remind ourselves skeletal muscle is sometimes called voluntary striated, okay? Cardiac muscle is sometimes called involuntary striated, and smooth muscle is sometimes called involuntary non-striated. Now, what I want you guys to pay attention to is also kind of where these things, what they kind of are, where they are, what they do, so to speak. The skeletal muscle being moving the skeleton, the cardiac muscle pumping your blood in your heart, and your smooth muscle being what changes the shape of hollow organs, for example, blood vessels and bladder and things like that. Now, what we're going to see is you're going to see a skeletal muscle, and when we look at characteristics, we're going to see it's very long, thin fibers. Okay, we remember talking about that. Remember that it had multiple nuclei for every cell, had more than one nuclei. We'll see why that was in this lecture, why that is the case. We're going to see that it is striated. We're going to see why those striations happen, so to speak. And then we're also going to see this idea that it is voluntarily controlled. Okay, we got to remember that. Now, cardiac muscle, involuntary striated, pumps your blood. Okay, we remember that. So it's got branched cells. Cells are branched in their structure. They have one nucleus. They are striated as well. They're going to have sarcomeres, etc. They're under involuntary control, and they have the intercalated discs on them so we can communicate from cell to cell. That'd be an important thing we talk about in A&P2 as we go through A&P2. Usually I teach an online class of A&P2 as well. I don't know if I'll be teaching it in the fall or not, which one I'll be teaching, so we'll see. Now, smooth is going to be slightly different. Again, we said it's in all these hollow organs. It's very spindle-shaped, single nuclei, non-striated, but it's also involuntary. So you will see that cardiac and smooth muscle are ones we can't control voluntarily. Okay. Now, here we can actually see them in those characteristics that we talked about in the previous slide. Okay, so let's talk skeletal muscle. From here on out, we're going to be focusing on skeletal muscle except for a little bit. I'm going to talk a little bit more about cardiac muscle, a little bit about smooth muscle, just a little bit later. But really what I want to focus on right here is our skeletal muscle. Now, our skeletal muscle, we're going to say that, it's mostly skeletal muscle tissue, okay? The tissue is mostly going to be skeletal muscle fibers, okay? That kind of stuff. And so what's going to happen is it's also going to have connective tissues. For example, dense regular connective connective tissues, dense irregular connective tissues will be attached to it and associated to it. Now we're going to see that the cell that's actually there is called a muscle fiber. Why is it called a muscle fiber? It's a very long, thin cell, so it's fibrous, okay? Now, skeletal muscles, they attach to bones. That's one of the big things about them. And when they do this, they can produce movement. That's one of the big things that it does. It produces movement. Skeletal muscles contract. When they contract, they pull. When they pull on tendons. And when they pull on tendons, they move bones. So we just talked about the joints and everything like that last class. And we talked about a few muscles in there. Specifically, I talked mostly about the rotator cuff and things like that. Now, they maintain posture and body's position. Tension of these muscles can actually maintain posture by pulling on the vertebrae and things like that. They support your soft tissues. They're going to have layers of muscle to make up your body wall. For example, everything is terrible. External oblique, internal oblique, transversus abdominis or transverse abdominis. I'm going to replace it here with transversus abdominis to get it to how we teach it now. because now we call it the transversus abdominis. So it guards body entrances, for example, the sphincters, the orbicularis oris, for example, the external anal sphincter, and those kinds of muscles like that. Okay, they control the openings of the things like your digestive system or your urinary system, for example. They maintain body temperature. They're going to be muscles that's actually muscles use energy, and when they use energy, that energy gets converted into body heat, and that heat warms our body up, maintain our body temperature. So that's why when you exercise, you warm up. You also can store nutrients. We can store glycogen and store protein in muscle. Muscle is a very big place to store glycogen. Okay. Now, we're going to start out and we're going to think about a muscle being made. If I pulled out a biceps brachii muscle out of the body, we're going to see that that is a whole muscle, okay? Biceps brachii, for example, rectus femoris, whatever muscle you want to pull out of the body, the whole muscle, the one that we identified, okay? One of those muscles that we identified is a whole muscle. Now, if we cut it open, we're going to see that inside of this is fascicles. These fascicles are bundles. And what are they bundles of? They are bundles of muscle fibers. And these muscle fibers, if we look inside of them, they're going to be made up of these little fibers called myofibrils, very small fibers that are inside the muscle fiber. And those myofibrils, if we zoom in on them, they're going to be made of sarcomeres. And these sarcomeres are going to be made up of myofilaments. You probably heard of thick and thin filaments before. That's a myofilament, a thick and a thin filament. So we have a whole muscle made up of fascicles. Fascicles are made up of muscle fibers. Muscle fibers are made of myofibrils. Myofibrils are made of sarcomeres, and sarcomeres are made of myofilaments. That is going from the biggest down to the smaller end of this particular thing, basically going down to the molecular scale, so to speak, okay? Now, what we're going to see is there's also going to be connective tissue layers, and there's three of them. There's one called the epimysium, perimysium, and endomysium. The epimysium upon the muscle, upon the whole muscle. This surrounds a whole muscle. Perimysium surrounds the fascicles. So it surrounds fascicles. Endomysium, internal, okay, surrounds muscle fibers. Epimysium upon the muscle, perimysium around the fascicles, endomysium around the muscle fiber, okay? Now, one very important thing that we're going to see is neurons are basically going to come in and attach to a muscle. They're going to basically be on it. There is going to be a very small space, just very small space that we're going to see. We're going to see the space between the neuron and the muscle is quite close, okay? But there's going to be basically they're in direct contact, so to speak, okay? And they're going to control the muscle contraction. So very important that we know that neurons, they're going to control the muscle contractions. They're going to tell the muscle when to contract, okay? Now, tendons, that's what's going to attach muscle to bone. So definitely a distinction we learned that was ligaments was bone to bone, tendon was muscle to bone. Now, one of the cool things, the epimysium, the perimysium, the endomysium, each one of these mycia, they contribute to the tendon. Their collagen fibers go into the tendon so that every muscle fiber pulls on the tendon. When every muscle fiber that contracts will pull on that tendon, okay? So, all right. So we could see the whole muscle. They took up, for example, maybe one of the extensor muscles or something they took. Okay, they take a whole muscle, and we see a whole muscle is made up of fascicles. The fascicles inside there are fascicles. Muscle fascicles are made of muscle fibers. Muscle fibers are made of myofibrils. Myofibrils are made of sarcomeres, and sarcomeres are made of thick and thin filaments that are called myofilaments. Okay, so we have a whole muscle made up of fascicles. Fascicles are made of muscle fibers. Muscle fibers are made of myofibrils. Myofibrils are made up of sarcomeres. Skeletal muscle, fascicle, muscle fiber, myofibril, sarcomere, myofilaments. Okay. Now, around the whole muscle is the epimysium. Around a fascicle is perimysium. And around a muscle fiber is endomysium. Okay. Now, muscles. Remember, muscles are attached at 2 points. They have origins, they have insertions. We learned this in lab. The muscles attach at 2 points, at least 2 points of attachment. Now, sometimes there's 2 origins, things like that. So there may be 2 origins and, multiple insertions and things like that. So there's going to be at least 2 points of attachment, at least. 1 origin, 1 insertion, at least, okay? The origin is the point of attachment where it does not produce movement when the muscle contracts, and insertion is the point of attachment muscle to bone where it does produce movement when the muscle attaches. So what we could say is origin is the point of attachment where when a muscle contracts, it does not change position. And insertion is the point of attachment of a muscle to a bone that when that muscle contracts, it does change position. And when that muscle changes the position of the structures, the movement that is made is called the action. That action is the movement produced by a muscle when it contracts. Now, between the origin and insertion, there's a central bulging part of the muscle. We call that a muscle belly. So when you look at the biceps brachii on somebody, on their arm, they have a large biceps, that swole muscle in the middle, the central bulging muscle, that is a muscle belly, okay? Now, skeletal muscles, a few things I want to talk about. Now, I said they're multinucleated. Now, why are they multinucleated? What's going to happen when they're embryonic? Now, they're a very large cell. They can reach about 12 inches in length, okay? So that's pretty long, a foot long, can reach about 12 inches, okay? So we're going to see they are multinucleated, okay? They're multinucleated. They contain multiple nuclei. And what we can see is when they're in an embryonic stage, there are these cells, these embryonic cells called myoblasts. And these myoblasts, they come together. And what this myoblast does is 1 myoblast, 2 myoblasts, 3 myoblasts, multiple myoblasts, they come together. Now, the nuclei that you see on a muscle fiber, when you see that there's multiple nuclei on a muscle fiber, that is, all those nuclei are the remnants of the myoblasts that came together to form it, okay? Now, there are some myoblasts that don't fuse together and don't differentiate. We call them myosatellite cells or satellite cell, okay? But the reason that a muscle fiber is multinucleated is all those myoblasts each gave us one of those nuclei. 1 myoblast, 1 nucleus. All the myoblasts that came together made the muscle fiber, okay? So it's a weird cell. Now, if we look at a muscle fiber, what we're going to basically see is we're going to see that it has a special name for its cell membrane. The cell membrane of a muscle fiber, because we're talking muscle, we call it a sarcolemma, which literally means flesh husk. Sarcolemma. Now, the sarcolemma, what's important about this is you're going to see the electrical impulses are going to generate on the sarcolemma, and they're going to travel along it. And we're going to see that's very important. Now, inside of a muscle fiber, there is a cytoplasm. Just like all cells have a cytoplasm inside, well, the cytoplasm of a muscle fiber is called the sarcoplasm. Basically, this is a flesh fluid, okay? Now, what we'll see is I want to be able to make the whole muscle contract at one time, a whole muscle cell to contract at one time. If I want to make the whole muscle cell contract at one time, what I've got to do is take my electrical impulse and carry it into the muscle cell. To do this, there are holes in the sarcolemma that travel down and extend down into the sarcoplasm, down into the muscle, and it takes these electrical impulses down deep into the muscle. These are called transverse tubules, parts of sarcolemma that extend down, that transverse, go down into the muscle. And these T tubules, we sometimes what we call them transverse tubules. Short for this is T tubules. Okay, we call them T tubules. Now, these T tubules will carry that electrical impulse down in the muscle and ultimately cause a muscle contraction. Okay, we're going to see this is very important. Now, myofibrils, we know they're inside of a muscle fiber, and so muscle fibers contain myofibrils. The myofibrils, what they're going to basically do is they're going to shorten, they're going to contract, they're going to get short, and when they get short, they're going to make the muscle cell get short. They are made up of proteins called myofilaments. Myofilaments come in 2 types, thick and thin and thick. Thin is made up of a protein called actin, and thick is made up of a protein called myosin. Myosin is thick, Actin is thin, and together these are called myofilaments. Okay, myosin is a thick filament, actin is a thin filament. Myosin is thick, actin is thin. Said myosin is thick, actin is thin, and together these are called myofilaments. Okay, these myofilaments, what they're going to do is they're going to shorten. Now, when a myofilament of a myofibril contracts, we pull the muscle cell, and the muscle cell pulls on the tendon. Do this with enough muscle cells, and you'll make a movement. Okay? All right. So let's understand more about a muscle cell. Inside of a muscle cell, there's an endoplasmic reticulum. There's smooth endoplasmic reticulum inside of a muscle cell. Because this is muscle, we call it sarcoplasmic reticulum. The muscle cell, the skeletal muscle cell, its endoplasmic reticulum, specifically the smooth endoplasmic reticulum, is called a sarcoplasmic reticulum. Now, there are very specific parts of the sarcoplasmic reticulum where the T-tubules pass between and bind to them. Where the T-tubules bind to sarcoplasmic reticulum, there are these chambers, enlargements of that sarcoplasmic reticulum called terminal cisternae. A cistern holds stuff. What the terminal cisternae do is they're going to hold calcium, okay? Concentrate calcium, as we're going to see. Now, if we take a structure that we have one T-tubule, one transverse tubule, and two terminal cisternae, we have what is called a triad. 1 plus 2 equals 3, triad. Now, what's going to happen is I like to think of the terminal cisternae is like a prison. And like a prison, we have prisoners. The prisoners would, if they were out of the prison, they would be causing tension in the society. So what we do is we keep these people locked up in prison. Calcium is the prisoner. We keep them locked up in the terminal cisternae, the prison. And like any prison, there's a guard. The guard is called calsequestrin. The ion pumps are going to be like the cops, arresting the calcium and putting them in prison. The calsequestrin is like the guard, keeps them in the prison. Okay? Terminal cisternae is going to be like the prison, where we concentrate calcium, the prisoners. The ion pumps are like the cops who arrest the prisoners and get them into prison. And the calsequestrin is like the guards that keep them in the prison. Okay, we want to keep calcium in the terminal cisternae when a muscle is not contracting. We're going to basically kind of think of it like, so think about it this way. If you found out that nearby where you live, that there was some prisoners who escaped prison, you might be very tense. You might be very concerned that some dangerous prisoners have escaped. Okay, there's a if I travel to certain places, it will say signs that will say if you're nearby a prison is like warning hitchhikers could be escaped convicts. So there's people who might try to escape prison, and you give them a ride and try to get away and escape. They might, so they might be dangerous. So what happens with our sarcolemma is there's these holes, and they travel down into the muscle. That is called a T-tubule. On both sides of the T-tubule are terminal cisternae. These terminal cisternae are going to concentrate calcium. Okay, electrical impulses are going to travel down into here and dump that calcium out and dump it out onto these areas right here, and that's going to cause a muscle contraction. We're going to see how this happens. Okay, it'll take us a little bit to get there. Now we're going to talk about a sarcomere. Now a sarcomere is the functional unit. This is where I say it's the functional unit. When I say it's the functional unit, this is the thing that allows the muscle to contract. It kind of works a little bit like an accordion. Okay, it shortens from both ends and then expands back out like an accordion. If you played an accordion, okay, it's gonna kind of do this accordion kind of thing. Okay, now myofibrils are made of thin and thick filaments actin and myosin. This is our functional unit. It's called a sarcomere. Now sarcomeres, it's going to be thin and thick filaments are going to interact. They're going to slide past each other, and they're going to cause muscle contractions. Now sarcomeres, they're going to have various things. They're going to have thick filaments, Thick filaments, these are going to be myosin, made up of myosin. Thin filaments made up of actin. Remember, myosin is thick, actin is thin. There's a protein that stabilizes thick and thin called titin. Then there's two proteins that have the same job. Now, let's think about it this way. Imagine that you have myosin, and myosin is a young man who wants to go on a date with actin. And myosin wants to hold hands with actin. But daddy and mama don't want their daughter actin holding hands with myosin. So mamas and daddies, they may try to prevent this young couple from holding hands. So troponin and tropomyosin are kind of like the parents that try to keep the young couple from holding hands. Actin and myosin. Myosin wants to hold actin's hand. Okay? Myosin wants to bind to actin. Troponin and tropomyosin's job is to prevent this from happening and regulate it. Allow it to happen only when it should happen and not allow it to happen when it should be, okay? So it's going to regulate muscle contractions, regulate the interactions between thick and thin filaments, troponin and tropomyosin. So it's going to be like the parents regulating whether or not these young couple can hold hands while the boyfriend, this young guy, is over visiting their daughter, okay? Now, we're going to look at a sarcomere. And what we're going to do with the sarcomere is we're going to take this sarcomere here and we're going to draw it out. And I should have pulled this up early, and dang it, I forgot to load this up before the video. I always forget to do something, it seems, and I forgot to load it up before the video started. And that is, I'm too far into it to start over. So I'm not going to start over. I just need to redo that. Okay. And I'm tied to this thing in so many different ways. Okay, so. And one of the things that's annoying, there we go, I want the top at the top. Okay, so let's start off. Let me move all this so I can actually move and be a little bit more free here. Okay. I'm going to move so I can sit in front of this and talk. Okay, so let's go over the sarcomere, okay? Now, the sarcomere, now, what I've got to do for this class is tell you that you will draw this for me for your 5 extra points. You're going to go and put this in a drop box for me. I'm going to make a drop box for you to put this in. So what I'll do is make sure you guys see that. There'll be a drop box using assignments tab on D2L. You'll draw this out and take a picture of it. Get a picture of it for me and take it and upload it. Okay, so this will help you guys because I will have this questions about this on the test, and I want you guys to practice on this. Okay, so we're going to start right here. Let me get that a little bit more straight up and down. So straight up and down the middle, we have the M line. Now this M line down the middle here, okay? Then attached to the M line, there will be several of these. And we're going to draw them however many, at least one more. There we go. All right, now we're going to draw these. What I want you guys to go in to do is to write thick. Write thick. That says thick. If you can't read it, sorry, I don't have fine tip markers. I only have these. I don't have the little fine tips that can write. That says thick. Let's see if I can write thick a little bit better here. Thick. Okay. Now, what we're going to do, now we're going to take this guy and we're going to go right here and we're going to draw this. Now, if you draw it out, you're going to internalize it better. And you're going to understand it, okay? You want to be able to draw this out. Now, on top of one of these, let's write thin. Thin. These are thin filaments. So purple thick, blue thin. All right. Now I'm going to come over here and I'm going to take my orange marker and I'm going to draw like this. Then I'm going to draw this. Like that. Those are more thin filaments. Now the orange, I'm going to write right here, Z line. Okay, that's a Z line. Over here, let's just write Z line as well. So we know that these are both Z lines. These are Z lines. Okay, so we've pretty much drawn most of it. What we're going to do, I'm going to take a green marker and I'm going to come inside here and I'm going to draw a squiggle like this from the inside to the tips here. Okay, like that, this little squiggly. Okay. And this is Titin. This is Titan. Titin, Titan. Okay, that's Titan. All right. So we have gotten the sarcomere drawn. Now what we got to do is identify the bands. Now there's various bands that we're going to see. So this is our sarcomere. Now what we're getting ready to talk about in lecture is there's an A band. A band contains thick and thin filaments, and I call it the dark band. A has dark. It is long as a thick filament. So the A band, what it's going to be, is all that right there. This is the A band. Okay. Now, notice in the A band, one of the things that we have is the M line. Okay. Now, inside of here, now you guys get it a little bit easier than my regular students do, my non-online. My regular students have to draw this on the test. They get to draw it during the test, but they have to draw it from memory. They don't get to just do it and then take a picture of it and send it. But they get to use it during the test, and so you guys will get to draw it too. But it's there's a kind of one loss there. This is the H band. H band. Okay. Now, next what we're going to see is a zone of overlap. The zone of overlap is found right here. This is our zone of overlap. Right here we have zone of overlap. Okay, zone of overlap. That's our zone of overlap, this space right in between the H band and A band. Okay, so our A band has our H band in it and our zone of overlap. Now, what is important here is our M line is the center of the A band. It connects our thick filaments together. Our H band is the lighter region on either side of the M line, contains only thick filaments. The zone of overlap is where thick and thin filaments overlap. Now, I band. I band is only thin filaments. I band there's your I band. Okay. There's your I band. Okay. Your I band. Now the I band is going to be containing the thin filaments. Now it goes from A band to the A band of another sarcomere. Okay, it's in between the A bands. The Z line bisects it. Sarcomeres technically go from Z line to Z line. Now, titin is an elastic protein that keeps in lines and allows sarcomere to snap back. It stabilizes. So it's stabilizing, and titin will be in the I band. Okay? So if we look at it, there is our sarcomere. Be able to draw it and take a picture of it. Send it to my assignments tab for the sarcomere drawing. I'll go ahead and open that up right after this recording. So thank you guys on that. Okay. So now, give me 1 second because I'm going to. So here's actually a drawing of it from the book. And give me 1 second because I'm going to need to get a paper towel. Okay. So sorry about that. I had to step away and grab something. So this is a drawing of the sarcomere here. Now, what we're going to do is, let me click on that and get back on here. So let's look at the thick and thin filaments, okay? So the thick and thin filaments, what we're going to look at is your thin filaments. They are going to be made up of 4 proteins. The proteins are made up of, one is F-actin, which stands for filamentous actin. It's 2 twisted strands of globular actin, or G-actin. The G-actins are going to have an active site for myosin to bind to. Remember, it's going to be like a date. Myosin wants to hold hands with actin, okay? Now, there's a glue. The glue that holds the 2 F-actin strands together is called nebulin. Nebulin is going to be like a glue to hold the 2 strands together. Now, remember these next 2, they are the regulators. They are the ones who want to control. They're like the parents who regulate the date. Okay, they are the people who supervise the date. They're the chaperones. Okay, you have tropomyosin. Now, tropomyosin here is going to be a lot like the dad, protecting G-actin's active site. Okay, basically kind of guarding the daughter. Okay, guarding his daughter. Covers the G-actin's active sites to prevent a muscle contraction. It prevents myosin from ever binding. Troponin has 3 subunits to it. Okay, troponin. So I always have to say there's troponin tropomyosin complex. And I always like to describe it really more like dad, and mama's more going to be like calcium. So we can think of the parents, and so I'll use different analogies to kind of help you guys understand it. But what's about troponin? There's 3 parts. One binds to tropomyosin, one binds to G-actin, and one binds to calcium. But when you put troponin and tropomyosin together, it makes what's called troponin tropomyosin complex. Okay. Now, thick filaments are made up of myosin. Myosin is thick, actin is thin. Myosin wants to hold hands with actin. How will it do that? Well, there's part of myosin, the fibrous part, called the myosin tail. Bundles of these make a thick filament. When you take bundles, like if I got a bundle of sticks, every stick is a bunch of myosin tails bundled together. Then what we have, though, is myosin heads, and they're the globular part of it that sticks out. Now, it's activated by ATP, and it binds to the active site of G-actin. Now, one of the things that's going to happen is we said that myosin wants to hold hands with G-actin, okay, wants to hold hands with actin. When they hold hands, we call this a cross bridge. Cross bridge is when they actin is bound to by myosin. When myosin heads bind to the active site of G-actin, we call that a cross bridge. Okay. Now, something that will be on the test, and this is where drawing the sarcomere will really help, is sliding fiber theory. Guaranteed to be on the test is what happens during sliding fiber theory. Now, according to sliding fiber theory, one of the things that happen is myosin and actin slide past each other. Okay. Basically, actin slides past myosin is a better way of saying this. Okay. Better way for me to say this, to be honest with you, than my notes kind of, because myosin is staying put. Actin is sliding past the myosin. Okay. Now, what happens is your H bands are going to get smaller, your I bands get smaller, your zone of overlap gets larger or increases, your Z lines go towards the M line or approach each other, and the A band doesn't change at all. Okay, so when you look at this, be imagining this sarcomere and what happens. Okay. Okay. So this is a sarcomere drawing of it with the different bands and everything as well. This is when it's relaxed. When it contracts, it slides. The Z lines approach each other towards the M line, and you can see what happens to the different things. You can see the H band gets smaller. You can see the zone of overlap increases. You can see the I band gets smaller, but the A band stays the same. Okay. All right. So that's our first major concept that we have to learn. Now that can be a little tricky. Get the sarcomere structure down, learn the sliding fiber theory. Basically, you figure out what happens. Basically, think about this working like an accordion, where the Z lines are being kind of brought together, okay? Smushed in together there, okay? All right, now what we're getting ready to talk about next is quite a difficult concept as well. It's called the neuromuscular junction. And that's where we're going to end today's lectures with the first really, and then I'll do some other complex concepts as well. There are a couple really complex concepts from this chapter, and I really want to talk about them. So we're going to start out with our neuromuscular junction. Now, the neuromuscular junction, neuron, muscular, muscle, junction come together. This is the place where we see neurons or nerves, the neuron of nerves, telling a muscle to contract. Remember, skeletal muscle contractions are voluntary. They're controlled by nerves. Nerves, like from the central nervous system off the brain and spinal cord, these nerves come off the central nervous system, and they're going to tell my muscle to work, okay? The NMJ is the place, the neuromuscular junction, or NMJ, is the place the magic happens. This is where the muscle gets told what to do. And it's a lot like a relationship where we got to have communication between the two parties, okay? Now I want to talk about like a marriage and kind of use a marriage as an example. So our neuromuscular junction is a synapse. Now what is a synapse? Synapses are nothing more, guys, than when two cells come together to communicate with each other. Okay. Now this is going to be a neuron and a muscle fiber. Now the neuron comes first. Neuron is the one that basically is asking the muscle cell to contract, well, telling it to contract. And so the neuron comes first. It's first in the sequence. So it is called presynaptic because it is first in the synapse. Muscle comes second. Because muscle comes second, it is second in the synapse, and it is called postsynaptic. Okay? All right. Now, when we look at the NMJ, what we're going to be able to see is the neuron is going to have an electrical signal. We call that an action potential. An action potential is the signal a nerve sends. Okay? It's kind of like what happens when a cable sends a electric current down the cable to a spark plug in your car to make the piston, make the cylinder fire and the piston move. Okay, so it's kind of imagine this axon, this neuron has an electrical signal traveling down the axon. It gets to the end, and there's synaptic terminals. The synaptic terminal is the place where the neurotransmitters are. And we're going to see that this neurotransmitter is called acetylcholine. Okay, acetylcholine. Now, acetylcholine is going to get dumped out into a gap found between them. Look right here. There is a gap, and this is called the synaptic cleft. Okay. Now I'm going to draw this out in a moment. We'll talk about it. Now there is a motor end plate where the motor end plate is heavily folded with junctional folds. These folds, called junctional folds that are indicative of the neuromuscular junction, are going to increase surface area for me to add receptors that can bind to these neurotransmitter acetylcholine molecules. Okay. Now, acetylcholine is the neurotransmitter. Now, it travels our synaptic cleft, and acetylcholine binds to receptors. Now, these receptors are a very special type. They're called nicotinic acetylcholine receptors. Now, nicotinic, what does nicotinic make us think of? Nicotine. It's excited by nicotine as well as acetylcholine. So it's nicotinic acetylcholine receptor because nicotine also stimulates it as well as acetylcholine. So we call them nicotinic acetylcholine receptors, or NACH receptors, okay? The nicotinic acetylcholine receptor. Now, nicotine also does it. What happens when acetylcholine binds is this will allow sodium to pass through, and the sodium passes through the protein, and this allows me to excite the muscle cell. This causes sodium cations, sodium ions, Na+, to enter and rush into the sarcoplasm. Now, there is an enzyme that breaks down acetylcholine called acetylcholine esterase, or ACHE. Now, what acetylcholine esterase does is breaks down acetylcholine and closes the gates. Now, a lot of pesticides affect acetylcholine esterase. A lot of your pesticides are acetylcholine esterase inhibitors. So this is why you will see the inability for a person who has been poisoned with some of these pesticides to relax their body. They won't be able to relax. We'll talk a little bit about why. Now, what we're going to do, let's go over the structure of that real quick, okay? And I'm going to write down up here, I'm going to write neuromuscular junction, NMJ, okay, NMJ. Now, the NMJ, what we're going to do is we're going to have our neuron, okay? And our muscle fiber, okay? Now, our neuron tells our muscle to contract, okay? Now, the neuron is called presynaptic, and the muscle is called postsynaptic. Now, inside this neuron here, we have what's called a synaptic terminal. There are these little vesicles, and these vesicles are filled with a chemical neurotransmitter called acetylcholine, ACH, acetylcholine, okay, ACH. Now, ACH, acetylcholine, is the neurotransmitter of the neuromuscular junction. Now, what's going to happen is, over here on this side, there's all these nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors, nicotinic NACH receptors. They are found in this gap right here called a synaptic cleft, okay? All right, so this little space. Now, the synaptic cleft is there, so we have space between these. You don't want them to touch. It'd be like shorting it out. What I always kind of say is it's like this, okay? In a relationship, you don't want to be smothering. You don't want to be just smothering, like, you know, and you also don't want to be too distant. You want to be just right. That is just right for diffusion to happen, okay? Now, what we're going to look at is some stuff going on. We're going to start off with, now there's an area here called the motor end plate with junctional folds. I'm not going to label it because I will get in the way of my drawing. And we're going to start out with resting potential. Now, resting potential, this is going to require the sodium-potassium ATPase pump. Now, what we're going to do is going to require the sodium-potassium ATPase pump. Now, just remember we have ICF sarcoplasm, and we have ECF, extracellular fluid. Okay? Now, there is this pump. And this pump, now we can say that it pumps out 3 sodium cations. Let me make it where we can actually read 3 Na plus and brings in 2K plus, okay, into the ICF. Now, what we said was it makes cells like salty bananas. The ICF, remember, is going to have a lot of potassium in it, and the ECF is going to have a lot of sodium in it. Now, the ICF has got less cations in, and the ECF has got more cations out. So the ICF is slightly negative, and the ECF is slightly positive. Let me erase that and make that look a little bit neater. There we go. A little bit positive. Now, let me make that a little bit clearer because I'm going to be able to adjust this in a minute. Okay, what we're going to do, there we go, really make that stand out. Okay, now, what we said was this cell is polarized. Okay, so this is resting. He's relaxed, not contracting, not doing anything, resting, relaxed, and all that, okay? Resting and ready to go for a muscle contraction. Now, what we're going to do with this is going to create a resting transmembrane potential, okay? The resting transmembrane potential references that the concentration of sodium out there to excite the cell later. There's a potential to excite it later. It's polarized, it's negative in, positive out. Okay, 2 potassiums in, 3 sodiums out. I said the other way. It doesn't really matter. At this level, it is more accurate to say sodium out, potassium in, because that is the cycle of the pump. If you ever have to know the pump cycle, actually how the pump works, I have a video where I discuss that on my YouTube channel that you're watching this on. Okay, so anyway. All right, so we're going to start off with neuromuscular junction acetylcholine. Now, acetylcholine is found in vesicles called acetylcholine. It's the neurotransmitter of the neuromuscular junction, okay? Now, the neurotransmitter of the neuromuscular junction called acetylcholine is a chemical that what it does is it's going to change the permeability of the muscle cell. It's going to change its permeability and other properties of the cell's plasma membrane. So that's what neurotransmitters do. Neurotransmitters, they change the permeability of a cell to things, and they change other properties of the cell's plasma membrane. So that's going to change and alter its permeability to sodium. There's an enzyme in the synaptic cleft called acetylcholine esterase, the yellow moons that are being shown on the screen. Now, we'll talk about that in just a minute. Now, what's going to happen at the NMJ is on the neuron, we're going to have an action potential come down the axon terminal. And that is a membrane change. And then what's going to happen is that is going to come down, and that charge is going to open up what is called voltage-gated calcium channels, and that's going to cause the release of the neurotransmitter acetylcholine. Now, let me show you how this happens. So what's going to happen is we're going to have down this, we're going to have an electrical potential right here and called electrical impulse called an action potential. Action potential. Now, this action potential, what this is going to do this is going to allow the opening of these voltage-gated calcium channels. Now, I'm going to have a hard time writing that in here. So I'm not going to write, but these are voltage-gated calcium channels. So they open with the voltage of an action potential. The electrical charge opens it up, and calcium is going to come in. Okay? And this calcium comes in. Now, I like to think of it this way. Now, think of these vesicles with acetylcholine as like prisoners. And we have now snuck a file in the cake so the prisoners can escape. what's going to happen is this calcium will allow these vesicles to be released, okay? Maybe it's kind of like we found some evidence that shows these prisoners are, in fact, innocent. And we give that evidence to the parole board, and the parole board says, let them go. They are innocent people. So what will happen is this will make the release of acetylcholine happen. So acetylcholine will dump out of the synaptic cleft into the synaptic cleft, and it'll cross it, and it'll bind to these receptors. Okay? So the ACH is released by exocytosis. Now, be able to put these events into order. Action potential rise, voltage-gated calcium channels open, exocytosis of acetylcholine. Be able to put that in order. That will probably be a test question. Have you actually order those particular events? Exocytosis happens. Why? The vesicles are made of the same stuff as the cell membrane, phospholipid bilayer. Now, what's going to happen when the acetylcholine binds to the receptors? This allows sodium to come in. And sodium is going to come in. And what's going to happen when this sodium comes in is this is going to let sodium enter the cell. Okay? Now, why? Why will sodium enter the cell? Number one, remember, there's high sodium out, low sodium in. Concentration gradient. Also, ICF is negative, sodium is cationic positive. Opposites attract. So what's going to happen is all this sodium comes in, and this cell gets quite positive. And when this cell gets positive, it's no longer polarized, it's been depolarized. Okay? It's depolarized. Now, this depolarization, when things depolarize, they get excited. And we're going to see what happens. Okay? Cell gets depolarized. Now, what's going to happen is, cell gets depolarized. Let me show you what's going to happen here, okay? I'm going to go ahead and explain it to you. Preview of coming events, okay? Preview of coming events is going to make this make more sense if I do it now. When this cell depolarizes, an action potential forms on the muscle cell, okay? And what happens, there was a T tubule and the two terminal cisternae, a triad. And this action potential goes down this T tubule and releases calcium out of that, and the muscle contracts. Okay? Now, that's to kind of put what we talked about into context. Okay? Now, know that happens. Understand that happens. This is going to help you put it together a little bit. But what I'm about to do, the cells depolarize and it's going to contract. Okay? Now, what I'm about to do is undo this. How can we undo this? We need an enzyme. And this enzyme is acetylcholine esterase, ACHE. And what acetylcholine esterase does is it gobbles up all these acetylcholine molecules. And what we're going to do is we gobble them up, and we stop bringing sodium in. Now, the cell is still positive. Okay? Now, what's going to happen is it stops making it any more positive. Now we got to undo it, make it negative. What we're going to do next, let's just pick right here. Let's just do it right there. We're going to let potassium gates open, and we're going to take a bunch of potassium out. And what's going to happen is the cell becomes negative again, and we call it repolarized. And what will happen is all that calcium goes back in, and the cell relaxes. Okay? And we say it repolarizes. All right? Now, this is a very lengthy and difficult to understand process. It will take time to get this down. Number one, I do have a video on this YouTube channel that goes through this, so you can watch it as many times as you like. That's what my best students have done, my former tutors and all the best students I've had. They've had to watch it multiple times until they just got it. It takes a while to click, but when it clicks, you understand it, and when you understand it, now I'm going to tell you something. There's a reason I teach it the way that I do. Now, I had a student that she was in nursing school, and she came to me, she said that, so she got into nursing school. She said, but the nursing school said, people, they passed the entrance exams to get into nursing school. They had the grades to get into nursing school. Not everybody had the knowledge to stay in nursing school. And what was happening, they were in pharmacology, one of the classes that wiped them out. And she said the class average for every test was in the 40s. And she was making in the 90s. And she said they were doing the drugs that dealt with acetylcholine. And she said everybody was just, they didn't understand it. They just didn't grasp it. They couldn't get it. She said, mostly just because you drew this and you explained this way and you went over it in the details you did. She said, I not only was getting A's on the exams, I was barely having to study because I knew this. I understood how the drugs worked. And she was acing the exams. And she was making it through. The thing was, she was understood the physiology. You have to know this to understand so many drugs, and it's got to be to this level. Okay? I'm not kidding when I say that. I do this for a reason. I help my students understand it to this level of depth for a reason. Okay? All right. Now, repolarization happens, and now part of this is because the positive ICF repels the potassium and gets pushed out, and it's also going from high to low. So there's two reasons why potassium goes out. All right, so this has all the events. One of the big things I'm going to have is what happens in a cholinergic synapse, what happens at the presynaptic end. Number one, action potential arrives. Number two, calcium channels open. Number three, the exocytosis of the neurotransmitter acetylcholine. Okay. That's the thing you're going to see on the test for sure, is those three events, okay? Now, what I'm going to basically do is end it with this and then pick it up with it again. And we'll do excitation, contraction, coupling, and talk about muscle contraction in the next lecture. So basically, we're going to say that what happens is, guys, when you the sodium ions go to the sarcolemma, when they come in the muscle fiber, they cause an action potential generated. That travels along your T-tubules. It goes down the T-tubules, and it gets to a triad. The triad releases calcium and triggers a contraction. And what this will require is that myosin heads have to be cocked. So kind of think of it this way. I'm going to use a gun as an analogy, especially a revolver, like a double-action revolver. Probably a gun you've seen at least used in movies or TVs. Maybe you shot one. I do enjoy shooting revolvers along with some other, firearms. But one of the things, if you've ever shot a double-action revolver, you have that really long trigger pull. If you pull the hammer back and go into single action, you have a short, crisp trigger pull, where it's easier to pull the trigger. Imagine pulling the hammer back and then the short trigger pull that you have. Okay, this requires our myosin heads to be cocked and loaded with ATP. And we're going to see how this happens in the next lecture. Okay, so the next lecture we're going to pick up. Please take time to learn this. It is tough, but you can do it, and I believe in you. Okay, I'll see you guys in the next one.